Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41-funded Mesoscale Mapping housed in the Martinos Center. It is our great pleasure to have Dr. Rita Schmid today giving us great BrainMap talk, and Marta from Martinos Center will introduce her to the audience.
Thank you. Yes, it's a pleasure to introduce Rita Schmid. She's a senior scientist in the Department of Brain Sciences from the Weizmann Institute of Science in Israel. And so during and after her master's in physics, Rita worked in a medtech company developing technology for non-invasive treatments by magnetic resonance guided focused ultrasound, including a neurosurgery device for treatment of tremor and Parkinson. After returning to academia, she did her PhD with Professor Lucio Friedman in the Weizmann Institute, and she developed an ultra-fast spatiotemporal encoding pulse sequence that can provide high robustness to magnetic field inhomogeneities. Rita then did her postdoc with Professor Webb at the Leiden University, developing metamaterials for 7 Tesla MRI. So since 2018, she leads a lab in the Weizmann Institute focusing on exploring new capabilities of ultra-high field human MRI, aiming to better understand the human brain function. And among her interests are methods of improving temporal resolution in fMRI and developing new contrast methods for 7 Tesla MRI. So today we have I see some music and dance. The title of our talk is Bebop and the Shuffle, exploiting frequencies to improve the measurement of long and short-term variations in the brain.
Okay, thank you for the introduction, and thank you for the opportunity to tell you about my recent two recent projects, where we want to explore the frequencies of the acquisition to improve the measurement of some potentially quantitative methods, and also a method for functional MRI that I will explain more about it. Okay. And before I will do it, I want to tell you a bit about my lab and Weizmann Institute. So we have a 7 Tesla Human MRI Center with the Terra system. Where starting from May 2019, we got our approval to scan humans, and our center includes pulse sequence developers and neurobiology researchers, and it includes topics like neurofeedback, smell lab, learning processes, and among them, my lab aims to combine methods to develop methods development and basic neuroscience. So my lab interest is to advance high-resolution brain functional studies, and among those topics, I'm interested to increase the effective temporal resolution in functional MRI, so we will be able to measure some short-term changes and to better characterize the neurovascular temporal responses, and also to learn about neural processes. So among those topics, I am interested to study functional MRI in especially challenging regions like the basal ganglia with high iron concentration. As a complementary method to study the brain function, we also want to measure long-term changes in order to characterize specific person. It's dependence, changes due to age, environmental effects, and so on. So we are interested in quantitative biomarkers. So in my lab, we are working to improve methods for fast 3D high-resolution T2 mapping at 7 Tesla, and also high spatial resolution spectroscopic imaging. And all those methods, we also want, of course, to increase the signal-to-noise ratio. So we also work on methods with metamaterial structure design, where we can improve RF transmission locally or RF reception. just in a glimpse, our recent works for quantitative biomarkers include methods that is based on a 3D steady state partial spoiled approach. Well, it was already shown by Scott Reader lab, and we continued its implementation for 7 Tesla, where we can have a phase signal dependence on T2 and the B1s, and so we can eventually reconstruct from such a 3D acquisition whole brain 3D T2 maps. And we also work on methods for high resolution spectroscopic imaging. So we developed a method that a variant of FC acquisition that allows us to achieve high resolution and to remove the constraint in FC variants of the spectral width limitation. On functional MRI temporal response, we are working on, first of all, to characterize what are exactly the effective temporal resolution that we have. So we are working with dynamic phantom that we got from, so you can see here the links for the company, and here Helmut knows much more about this phantom. So what we do is we generate dynamic signal changes that are shifted between them in time, and we repeat those experiments many times, and we can measure and examine different protocols and their parameters and what is really the effective temporal resolution that one can get. And then working on more close to some neuroscience studies. We want to examine the basal ganglia, where we are running functional MRI experiments with motor task of some complexity of finger tapping, and we are checking whether what are the differences between when the volunteer performs the task and when the volunteer observes the task shown in the video, and we see interesting activation in the basal ganglia that we are now exploring. And as complementary to all of those methods, we want to, as I said, methods that will improve the signal or reduce the SDR. So to examine all of those, we let's say continue the development of 3D HL phantom that also based on the work here from Larry, where we added a brain mimicking metabolites to the phantom and also added the lipid layer, so we can examine all of those methods that we are developing at 7 Tesla. And we also, for example, examine how to reduce the power deposition in functional MRI scans when we are talking about high resolution. We also, usually it means that we will need many slices to acquire, so it means high SDR in 7 Tesla. So we worked on methods where you will not need to use fat suppression, and only based on the channel receive sensitivities similar to other acceleration methods, we can also remove the signal of the fat. And finally, I will mention the metamaterial designs which we can use to improve the signal. So now, finally, to the topics that I want to talk today. And so those are the two topics. One is originally based on the fact that we want to acquire high resolution images and then artifacts like you see here in this gradient echo sequence become highly important, because we really want to see the local information, anatomical information in high resolution. So any such small artifact can become important. So we work here on a method how we can remove such artifacts. And again, the other method will become soon more clear what exactly we are talking about. So in this method, we developed a sequence which generates an auditory stimuli within the sequence itself. Okay, so for the first topic, so like I showed you before, so our problem is one of the major ones is the pulsation artifact, but it can be also due to breathing. And for example, here you see the eye movement. So any artifacts that are coming from some physiological signals that have fluctuations in specific frequencies, and they will generate artifacts, local artifacts like that. Here, another example that you see with partial RF spoiling, where in this case, the ventricles generate, again, due to cardiac pulsation, significant artifact. So one of the ways to solve it, which we were thinking about, is to randomize the acquisition of the k-space order. Okay? And it turns out that this is not a new idea. It was already proposed by Hake and Patrick in 1986, probably as most of our new ideas that we are thinking about. So how it can work. So here you see the simulation in 1D. So what you see is the standard acquisition. It's only just the ordered one. And let's say that we have our object signal which we acquire in time. And in case there is no physiological noise, we will get our image. If we have some physiological noise that oscillates in specific frequency, it will add up to our signal, and we will get, after Fourier transform, we will get an artifact that you can see here in an image. If there are a range of frequencies or also coming from different locations, we will have a set of such lines or strips artifacts that we saw in our previous case. So what will happen if we will scramble or randomize the acquisition order? You can see here that our signal was kind of scrambled, but the physiological signal is still the same because it requires in time. And so now we will reorder it back. So our signal is exactly as we wanted, but the physiological noise became really like a smeared noise signal. And this is what will happen in our image, that we get rid of the artifact. So it sounds great and a good solution. So we went to the scanner to test it, and it turns out that it works, but not always. In some cases, so this is what. So in some cases, it works well. What you see here on top of the phantom example, and the bottom is the in vivo example. But at some frequencies, at some bandwidths of the readout, even on phantom, where there is no physiological noise, we got a significant increase in the global noise in the image. And this similar happened in some cases in our scans in vivo. So we assume that in phantom it's probably due to some eddy currents, because now we are jumping in our k-space in the phasing cone direction, so we can have very significant jumps. And in vivo, it can be due to some slow global movement. So all this movement becomes converted to noise. So we want to generate a trajectory that will avoid large phasing cone jumps and will filter slow changes. So in order to examine what will work well, we prepare the simulation. Where all you see in our simulation is our physiological source. So this is the signal that is expected to be the artifact. So we can plot the signal intensity as function of the frequency. And so what you see here is its behavior normalized and we now can compare other trajectories and how the signal, how the artifact can be removed. So what we see now is 2D acquisitions. This is a two because we have two phase encoding directions, and the plot shows just in colors the order. So this is a matrix of 128 by 128 points, and you see the colors that go up to some eventually to the edge. You can also see here a zoom in. So we acquire line by line, and we have connection between the lines. And at the bottom, you see the time shift histogram compared to the time of the acquisition in the ordered case. Therefore, all of those, the time shift histogram here is everything is at 0 because this is the order case. So now we finally can compare different trajectories. So this is what happens in a fully randomized example. You can see the randomization. You can see the histogram that now it shows the distribution of the time shift. And you can also see that the artifact, we reduced it significantly by huge factors, but it is very sharp. So this is our problem, that we want to have some filtering effect. We want to not touch the slow movements. So first we examined some semi-randomized filling curves. One of them is generalized Hilbert curve, which what's interesting about it, that it has small jumps in phase encoding dimension, but it also generates this distribution of the time shift, and you can see that it improves significantly, it's reduced significantly the artifact, not as good as full randomization, but the problem that it also doesn't have the filtering properties. So then we came up with an idea that we can, so what you see here, there are two signals, one that changes slow, one with low frequency and one with higher frequency. So what we can do is to locally scramble signals, defining some segments. So in each segment we scramble, okay? So what you see that for this signal we scrambled it totally, so we removed it. But for the slow changing signal, we didn't touch it, so we still can see it, okay? So this is what we did. We implemented it with local scrambling. What you see here, the segment scrambling. Okay, you can see inside, if you zoom in, that there are some patches so that inside it's scrambled, but otherwise it goes similar to the order one. And you can see now that we define some cutoff frequency, and therefore above this frequency, the artifact goes down, and below this frequency, we can still get the signal. So we will not touch slow global movements. But you can also see that it has some kind of side bands. And so we wanted to remove also the side bands, and therefore we came up with another step where we perform this similar scrambling, but without clear edges between the segments. And therefore now we have what we aimed for. So above the cutoff frequency, it behaves like a full scrambling, and below the cutoff frequency, we don't touch the signals. Okay, so now we examine it in the real in vivo scanning, and we started with a cutoff frequency of 1 divided by 6. So we expected it to cover well cardiac and breathing frequencies. So in most of the cases it worked, but not in all cases. So we went to much lower cutoff frequency and set it to 1 divided by 20, and then it worked. So we were happy about it and continued to examine it. So now going back two examples. Now, also on Phantom, the images look well, the same noise as the ordered one, and also those images are clean and there is no global noise that is added. And now, in order to examine in fair conditions in vivo scan, we wanted to examine it at the same, to scan in the same times simultaneously the ordered and the scrambled acquisition. So we implemented a sequence where we are interleaving the case-based acquisition. So we will have eventually both the ordered and the other trajectories that we want to examine. So this scan will take twice long, but this is only for comparison purposes. So here you can see the comparison. You can see on the left side, those are the ordered, the interleaved acquisitions. On the left side, you see the ordered scan, and on the right side, one of the trajectories that we examined. I'm not sure that you can see well the artifacts, but in general, in the order case, you can see the artifacts, the strips in the image, and in the local scramble to be don't see. You can, at least this one for the eyes, you can clearly see, I hope. And in general, the other areas are also clean. And for the Gilbert case, it's relatively clean, removed the artifact, but there are still some small remaining artifacts that are there. It's again supports the simulation that we saw, and in this example, the full scrambling also worked well and cleaned the artifact. Here another example, this is only one scan, the same scan, interleaved scan with ordered and local scrambling, just three different slices. And so you can see that it's not only coming from large blood vessels, the artifacts are also in some different regions, and even in small, again, I don't know if you can see, but there are some tiny stripes also in, yes.
And this is 3D, right?
Yes.
Does the scrambling affect your choice of color?
So we ran, so those scans were with grappa for, in our case, factor 2, but so we didn't touch anything about the grappa, only the order. But in general, it is the next step that we can maybe also take it into account and improve it some. But at this point, just as is, only the order. Yeah, exactly. Yeah, so we sample the whatever grappa plan to sample, but in order that with the local scrambling. And also you can see, I'm not sure that you can see, but there are kind of black lines here. So those we think that due to breathing artifacts, and those are also removed in our local shuffling. And here, the other example, it's another scan. It's a scan with the partial, it is scan which is part of our quantitative tissue mapping method. In one of the scans, we have a significant artifact near the ventricles, and this one, again, almost totally removed. And recently, we also examined it in for angiography for top sequences. This is really a very preliminary result, so still not a full examination, but we also can see in the ordered case that those artifacts are clean, and we have initial feeling that maybe we can get better delineation of the vessels, but this requires further analysis. So to summarize this part of the talk, and local scrambling in general, in contrast to full randomization, it is frequency selective, So it should not affect slow global changes. And we have also smaller phase encoding steps, so there should be less eddy current effect. And so it should provide significant artifact reduction for T2 star, for SWI sequences, which are based on RF spoiled SSFP, but it also can be useful for other 3D acquisitions. In general, for this method specifically, there is a benefit for 3D acquisition because short TRs, it provides wide range of frequencies that we can cover. The extra phase encoding dimension has more phase encoding points, so it allows better scrambling effect. And in general, the effect of the artifact or the improvements depends on the artifact source size compared to the full field of view. So since we have 2 dimensions, it's again proportional. The artifact size becomes much smaller compared to the full field of view. But we are working on ideas how to do it also in 2D case. So now moving on to somewhat different topic. The second topic is so where we started to think how we would like to target high precision functional MRI. And thinking about it, discussing about it in my lab, we came up with the idea that audio tasks can be a good candidate to generate short stimuli with high repetition rate. However, it is well known that existing acoustic noise generated by the acquisition gradients is a challenge in auditory stimuli. So we were thinking how to deal with it, and we came up with the thought that we can use the sequence itself and to generate the stimuli, the auditory stimuli. This way we can have high built-in accuracy. This is the accuracy of the MRI acquisition. And there is no need to synchronize with the scan with any external tools. So we implemented a sequence, EPI sequence, where we alternate the sound using changing the echo spacing of each acquisition. Okay, so echo spacing, it's time to acquire a single line, which defines the rate of the acquisition. Okay, so now let's see if I can. Okay, so the regular sequence. This is again specific parameters, of course. So this is the regular API acquisition. And now our acquisition. In our acquisition, so we set some blocks where you will hear fixed echo spacing, the regular acquisition. And then the changing sounds, just by changing the echo spacing of the API itself. And then it goes back again to the fixed echo spacing. And then playing with the sound. So at this point, it's still, so we called it Bebop for beeping ball pulse sequence, but it's still far from jazz music, but maybe one day. So we have changes in the signal. So yeah, so this is what I will show in a minute. Yeah. So this is how it is arranged. So what you see here, the slices and the time slices in y-axis and time in x-axis. And so we have our fixed pitch block, so the echo spacing in the same, and then we have the randomized pitch block. So we have changing echo spacing in a range of, so we don't want to change it too much. So here it's from 0.5 to 0.8. And this is how it looks as, let's say, the main expected sound frequency. Okay, so we have again the fixed and then the randomized block. And this is the recorded sound. So again, here the acoustics amplitude, how it looks like. However, when we examine the signals, we saw that in different regions, when we change the echo spacing, the signal also changes, not a lot, but let's say significant enough for functional MRI. So we needed to deal with it. So temporarily, for a temporal solution, what we did is we choose a subset of slices that we define that we are really interested to examine, and in those slices, the echo spacing is fixed as the original one, and all the rest are changing. So you can see it here that there are some points that go back to the fixed echo spacing, but it's since we jump all the time, also the interleaving of the slices helps, so we jump enough. As you heard, it still sounds like a randomized enough sound. And so now we can check how the signal looks like. So here we see an example of 12 slices, and here we choose to keep a fixed echo spacing in 4 central slices. And if we zoom in the slice that we didn't keep fixed, so there are, first of all, changes due to the B0 in homogeneity. Okay, because so we expect it due to longer acquisition time, and in slices that we kept, the signal is as we expect. There are some fluctuation, but it's the regular changes. So now we can examine it in region where so we define our block design or the hemodynamic response that we expect based on the fixed echo spacing is our reference block, and the randomized pitch is our task block. And here you can see voxels signal it in voxels with t-test above 2.5, and you can see that if we compare it to the hemodynamic response, it looks it fits very well. Again, in slices that we kept with fixed echo spacing, but if we look in slices that we didn't keep, you can see that there are some changes, and most importantly, the changes are not at the same time as they don't correspond to the hemodynamic response. They happen exactly when we change the echo spacing. Okay, so we can, in general, maybe we can also analyze it retrospectively and correct for both things. So now we run a few experiments. So this is an experiment with results that I showed you before with the signal change. So here we see that our 4 central slices are in the auditory cortex, and we see very well the auditory cortex voxels. And we can also run an experiment in which we say to the volunteer, when you hear a randomized pitch sound, then tap with your fingers. And in this scan, for example, we choose that we will have 3 slices in the motor cortex and 2 slices in the auditory cortex, and we see the activation in the relevant regions in the motor and the auditory cortex. So now, as the next step, which we are just starting, we want to examine and to study the different regions that are involved when we compare the regions that are activated in case that the person listen only, and where we combine the auditory and motor task. And you can see what we were glad to see, that there are very high signals in the basal ganglia, which we are really interested to explore. And so this is our next step, and we also want to examine the different temporal responses between the different regions. starting from the auditory cortex, basal ganglia, and primary motor cortex, and so on. So those are still highly preliminary results, and we need to continue the analysis and to generate maybe more better defined experiments. As the next step, so this is only a recording of the auditory signals. What we want is also to examine the activations in the brain from what we actually started is that we want a short stimuli and with high repetition rate. So here just the recordings of signals where we change the echo spacing, and you can see that the stimuli that we expect has different energy, let's say. So we want to be able to see what kind of effect it will have on the activated region. And here what you see is, for example, multi-echo recordings of the acoustics, and we can get different sizes of the stimuli when we are talking of sizes of very short stimuli. But this is again as the next steps of our study. So to summarize this point, we propose a new approach to generate an auditory stimuli by controlling the acoustics of the particles themselves. We showed a feasibility study with a block design, but there are still challenges with many eddy current effects that affect the signal in some of the slices. So in future work, the aim is to also to separate to be able to correct for the eddy currents or to separate the functional responses from the eddy currents. And so this is an opportunity to define a new set of experiments which can provide insights into both the auditory as well as neural network processes. And so I want to then, so both of those projects were a joint work with Amir Segener. And I want to thank also to the Weizmann MRI Center support, and this is my lab. And thank you for your attention.